This is what we're going to talk about today. The title of my talk is The Human Body, Evidence for a Designer. Many of you, I'm sure, are familiar with this painting, Michelangelo. It's called The Creation of Adam. It's in the Sistine Chapel, of course, in Vatican City. And certainly it's an artistic representation of God's creation of the first man, right? Adam. And it portrays, I believe, and most people believe, God's direct involvement. His fingers essentially on the creation of humanity. Adam, or Adam in Hebrew, is also used not just to describe the individual Adam, but also mankind, Adam, or humanity. So that's what we're going to talk about today. The science of the human body. Does it point to a designer? And potentially a creator's design. So, of course, very relevant to this talk, Psalms 139. For you created my innermost being. You knit me together in my mother's womb. I praise you because I was fearfully and wonderfully made. Your works are wonderful. I know that full well. So, knit together, fearfully and wonderfully made. I'm going to try and show you some of those details of being knit together by looking at the human body, its structure and its function, and some of the elegant physiology and control systems that maintain the life of the human body. I hope to provide you with some good sort of foundational information and hopefully some interesting and compelling evidence so that when you walk away with intellectual honesty, you might agree with me that the human body is best explained by biological design. So this is the debate, right? Debate, is the human body designed or a product of natural or materialistic processes? This isn't a recent debate. It goes back to about the 5th century BC. And in those times, there were philosophers like Democritus. He'd be in the naturalistic camp. It was called atomism at the time. And then there was Plato and Aristotle. They were more in the design camp. They felt like there was this transcendent mind in their philosophy that had a direct part in what we see in nature. So this is very similar to today's debate, where there's naturalism or evolution versus the idea of intelligent design. So just to clarify some terminology, and you'll hear me use this during the talk, and I don't want to confuse anyone, but an evolutionist can also be referred to as a materialist or a naturalist. Material causes, natural causes defined by evolution, created all life, including the human body. Intelligent design posits that there was a designer involved in nature and life, including humanity. So historically, William Paley is given sort of credit for creating this intelligent design perspective. He wrote this book called Natural Theology, and he said this: I take my stand in human anatomy, the necessity in each particular case of an intelligent designing mind for the contriving and determining of the forms which organized bodies bear. Certainly not the language we would use today, but nonetheless, wherever we see marks of contrivance, design, we are led for its cause to an intelligent author. The marks of design are too strong to be gotten over. Design must have a designer. In his writings, he presents this idea, which many people quote, this watchmaker analogy. He describes basically finding a watch when he's walking. I can't remember if it was on a beach or on the ground. Anyway, he finds this watch, and the characteristics and complexity of its parts implied the work to him of an intelligent designer, and no one would refute that. So by analogy, Paley asserts that just as the watch requires a watchmaker, so too complex life requires a designer. Then came Charles Darwin, you know his name? Yes. He's credited, of course, with the theory of evolution, and in 1859 he wrote a book called The Origin of Species. So he said this: all species of organisms arise and develop through the natural selection of small inherited variations that increase the individual's ability to compete, survive, and reproduce. So that's survival of the fittest, right? We know that as the Darwinian theory. So to him, life is a product of chance events. Now we know them as mutations and natural selection. So what he does is he says that nature sort of molds, nature selects, nature creates. natural selection becomes the substitute for a designer or a creator. So to clarify a little more terminology, Darwinian evolution, now we know it is neo-Darwinian evolution. I presented this slide at a church, not on this topic, not long ago, and the guy who invited me, out in the middle of my talk, he goes, "I'm becoming a pig." So here's what neo-Darwinian evolution is. Natural processes, random genetic mutations, and natural selection alone create life's diversity, survival of the fittest. All life results solely from undirected transformations of an original life form. Evolution is the creative force of life. So the neo-Darwin evolutionary perspective is, and you'll hear me use these terms, and you'll hear some of the authors that I quote use these terms. So evolution can be described as, in the scientific literature, the standard evolutionary theory, or standard evolutionary synthesis, or modern synthesis. So if you see those terms, that's what they're talking about, is the Darwinian evolutionary theory. So for many Christians, the theory of evolution represents a real threat, right? It claims that unguided, undirected, and biological processes created life and caused those life forms to undergo transformations, generating the diversity of life that we see throughout Earth's history, including mankind. So this is in contrast, of course, to many Christians or theists that hold that a creator brought life into existence and orchestrated life's history for his purposes. Michael Denton said this about Darwin's theory in his book, The Miracle of Man, and its impact on cultural as well as scientific thinking. He says, Darwin argued that the only causal factor responsible for the origin of our many planet species was the blind mechanism of natural selection working on chance variations. We, humanity, too, were the product of blind forces that did not have us in mind, for the simple reason that nature, being mindless, had nothing at all in mind. All notions which placed humankind in any special or privileged place were banished from mainstream academic debate. So Darwin has defined how many people in the world look at life, including humanity. And this, in my opinion, has had profound moral, ethical, and theological ramifications. So now let's look at this design argument. Teleology, it comes from the Greek word teleos, which means purpose and or goal. So the teleological argument is basically that it's the perspective that purpose and design are a part of and are apparent in nature. So the modern design argument frequently focuses on this idea of purpose and design. In standard evolutionary theory, there is no purpose or design in nature. It's random events. Design and purpose requires what? An intelligence or a mind, which would point to something beyond natural causes. So by fiat, standard evolutionary theory stands in opposition. Teleology, again, is the foundation of the intelligent design perspective, and there is design and they claim that there is design and purpose in life, in biology, and including the human body. So intelligent design, here's what they say. It holds that there are telltale features of life that are best explained by an intelligent cause. Life is here as a result of the purposeful action of an intelligent designer. Historically, the intelligent design perspective has been sort of pushed aside by the scientific community or looked down upon by many evolutionary biologists, poo-pooed, if you will. Interestingly, and we're going to ferret this out as the talk goes along, but in the last 10 to 15 years, intelligent design scientists, through research and publications, have presented some significant challenges to standard evolutionary theory that I think can no longer be ignored. And they have truly forced, I think, evolutionary biologists to sort of rethink, and you'll see they are rethinking standard evolutionary theory. Again, we'll discuss this. So this is a quote from Richard Dawkins, evolutionary biologist and atheist, arguing against design and sort of making fun of Paley's watchmaker analogy, right, with the title of his book, The Blind Watchmaker. He says this: biology is the study of complicated things that give the appearance of having been designed for a purpose. So even the atheist, the evolutionary biologist, even to him, the complexity of life appears designed for a purpose, but he claims it's not. Francis Crick, remember Watson and Crick discovered DNA in the past? He wrote this book called What Mad Pursuit. He makes a similar statement. Biologists must constantly remind themselves that what they see was not designed, but evolved. So you see this sort of presumptive bias. It looks designed for a purpose, but to Crick, it's not. So determining intelligence. This is a little bit of a big challenge for the intelligent design perspective. Science certainly possesses the capacity to investigate intelligent causes. Archaeologists, anthropologists, for example, they study artifacts and infer intelligent causes versus natural causes. Forensic science does the same thing. It tries to tease apart sort of purposeful or intelligent causes versus accidental or natural causes. So determining intelligence is sometimes referred to as inferential science or evidence for the best explanation. Stephen Meyer said it this way. He's a Discovery Institute guy, very well-spoken apologist and incredible writer. He said this in this book, Signature in the Cell. He said, by intelligent design, I mean the deliberate choice of a conscious intelligent agent or person to affect a particular outcome and or objective. So the implication, of course, here is that intelligent agent could be God. And so that makes many in the scientific community stand up and object. So that's some sort of informational background that I think is relevant to our conversations moving forward. So I'm sure you're thinking, is he about done? No, that's just the intro. But it's important, I think, for what we're going to discuss. But first, I just want to, as an aside, most physicians, including myself, move through our educational experience trying to learn and understand the principles of medicine, anatomy, physiology, biochemistry, pathology, pharmacology, all of this subject matter. We usually didn't contemplate the idea of design in the human body at the time we were being educated. Now, having been raised a Christian and certainly developing my own sort of personal faith and perspectives, I always accepted the premise of design, but certainly the way contemporary science and medicine is taught, such an idea is not a part of the conversation. So I'm a cardiac anesthesiologist. I like this slide. What's an anesthesiologist? Anesthesiologists are doctors who keep patients alive while surgeons do things that would otherwise kill them. And I think that's valid. I actually intended to show you some educational videos about cardiac anesthesia that I use to educate anesthetists, students that help anesthesiologists. They're like nurse practitioners, PAs for anesthesiologists. But I didn't have enough time to do that. And John said, you cannot talk that long. So before I discuss some sort of human physiology, we need to differentiate between sort of single-celled life and complex multicellular life, like the human body. Single-celled organisms like bacteria, for example, they get what they need. They get oxygen and nutrients from the environment that they're in contact with, because their cell is in contact with the environment. The human body, on the other hand, is a complex multicellular life form, roughly 40 trillion cells in the human body. Most of our cells are not in direct contact with the external environment. Yet, like the single-celled organisms, each of those cells needs its nutrients. It needs its oxygen. It needs electrolytes, and it needs to get that all from the environment. It's a supply chain issue, right? A contemporary term. The human body needs to have systems in place to get what its cells need from the environment. So how does it do this? The human body is made up of organ systems, at least we define it that way. Complex biological systems accomplish these tasks. So they divide the organ systems into 11. The nervous system, integumentary system, which is your skin, respiratory, digestive, excretory, skeletal, muscular, circulatory, endocrine, reproductive, and immune. So that's the 11 organ systems that we in medicine and physiology talk about. These organ systems are frequently described as sort of separate, as you can see in that slide on the left, but in reality, they are interdependent with one another, and they cross over and affect one another in amazing and surprising ways. So they accomplish physiologic tasks not on their own, but integrating with other organ systems. And it's incredibly fine-tuned in order for our body to maintain life. So I'm going to focus on just a few, because we don't have enough time, John said, just a few physiologic systems which I hope provide you with an appreciation for their amazing complexity and elegant ways that they function. So ABC, anyone ever heard this acronym in medicine, or CPR, basic life support? ABC, airway, breathing, circulation, right? So, and it relates, all these relate. They're fundamental to life, right? And they all relate to what? Delivery of oxygen to our body's cells. Oxygen is necessary for life. It's required by all 40 trillion of our cells. Oxygen sort of has primacy status because unlike water or food, which our bodies can store, we need a continuous supply of oxygen. Even if our body is deprived of that oxygen for just a brief period of time, let's say 4 to 6 minutes, we'll either die or we'll suffer severe damage. So how does the body deliver the oxygen to its tissues? Well, blood, right? So whole blood, blood that comes out of your veins or is in a bag that I transfuse to patients. Actually, these are packed cells. But blood in general has a multitude of things in it. It's got red blood cells, white blood cells, platelets, proteins, sugars, hormones, salts, coagulation factors. There's a lot of stuff in there. But again, we're going to focus on red blood cells, and I'm going to call them RBCs, right? So when you hear me say RBC, I'm meaning red blood cells. And they're the cells which, again, carry the oxygen. So RBCs are these biconcave cells. You can see them there on the lower right. It's like a donut with the center not out of it, or better yet, a kolache, which I see over there, right? That has that indentation in the middle. So we've experienced both of those. But basically that conformation, that shape is important because it increases the surface area of the red blood cell and allows for better oxygen transport. There are roughly 20 trillion RBCs in the human body. So we have more red blood cells than any other type of cell in the body. I didn't know if you put together, I said 40 trillion cells in the body total. 20 trillion of them are red blood cells. Tells you how much blood we got, right? Now, RBCs are smaller than many of the other cells. They don't necessarily take up the same mass, but as far as number of cells, they make up essentially half our body. So that's a lot of red blood cells. So why do our cells need oxygen? Oxygen is required for our cells to produce energy that each of those cells need to carry out the functions they need to perform. And so it's like this. I was told to point at things because otherwise. So essentially, we need glucose. We're not going to talk about glucose, but oxygen is needed. That comes in through the red blood cells. This is called cellular respiration. Those two compounds, or chemicals, basically produce ATP, which is the chemical that is used to create energy in our cells, and a byproduct called CO2. We all know about carbon dioxide, right? I'm not going to talk about climate change. Essentially, this is the chemical reaction, respiratory or cellular respiration, that produces the energy and that oxygen is needed for. And all of our cells need oxygen. Brain cells, muscle cells, liver cells, kidney cells, they all need oxygen to produce the energy they need to perform the tasks that they have to do. And we'll discuss this more, but I just want to point out a couple things. But you can see here in this slide, and we're going to discuss some physiology about this, but essentially our blood, our RBCs, right, carry both oxygen, this is tissue here, or tissue here, it carries oxygen through the blood vessel, these are called capillaries, where the oxygen is released to the tissue, and the carbon dioxide, that byproduct, is put back into the blood and carried by red blood cells back to the lungs, where we what? Exhale and eliminate it. I see some doctors in here, and they're going to go, now you said that wrong. Again, we'll discuss this a little bit more in the future, but here's some scriptures that I think give sort of theological support for the importance of blood. From Leviticus 17:11, for the life of the flesh is in the blood. From Deuteronomy 12:23, the blood is the life. And from the New Testament, Ephesians, in him we have, him, Christ, we have redemption, life through his blood. So, here we go, some physiology. So back to ABC, right? We're going to cover A and B, airway and breathing. So let's take a quick look at the respiratory system. This is the initial step in bringing oxygen into the body, right? Where do we get the oxygen? Well, we get it from air. Air is 21% oxygen. So we need to bring that air into our lungs to get that oxygen to our tissues. When we breathe in, it first enters through the nose or mouth, then enters the larynx or voice box area, if you will, then into the trachea. Am I coming across okay? I think when I turn my head. Can you hear me all right? Yeah. Okay, perfect. Then down into the trachea, which essentially goes into the lungs. You have two main stem bronchi, the left and right, that then split into what are called bronchioles, that are smaller airway tubes, that then end in these little tiny sacs called alveoli. So this is a good picture. Air comes down, trachea, bronchi, bronchioles, and ends in these little teeny air sacs that are surrounded with teeny little vessels called capillaries. And these air sacs, this is where the transmission of both oxygen and CO2 takes place. So oxygen is delivered to the blood in these alveoli, CO2 is taken up into the alveoli from the blood, and then exhaled. Oxygen comes in to the blood, CO2 goes out, exhaled, okay? So here's a sort of cartoon of, again, what I just described takes place. Air comes in, this is the alveoli sac in the lungs. Oxygen is delivered to the red blood cells and then transferred through the vascular system into the body. CO2 comes out of the red blood cells and is exhaled. Here's a microscopic image of those little alveoli that we have in our lungs. So this would be a bronchiole, this big tube right here, but it branches into these little alveoli, which again are the air spaces where oxygen and CO2 are transferred. Terminology. So you're going to hear me use the word deoxygenated blood and oxygenated blood. So when I use that, keep in mind that it makes sense, right? Oxygenated blood is the stuff, the blood that's picked up oxygen. Deoxygenated blood is the stuff that's lost its oxygen to the tissue and now is delivering CO2 back. Deoxygenated, oxygenated. So again, ABCs, airway, breathing, circulation. Now we're going to go to C, a quick run through in human physiology, right? How do we get the RBCs carrying that oxygen to the tissues? Well, it starts with the heart, right? Here's some interesting facts about the heart. The heart beats 100,000 times a day, 40 million times per year. In an average lifespan, your heart beats 2.5 to 3 billion times and pumps 60 million gallons of blood, never stopping for rest. If it does stop for rest, you're going to rest a while. Just some anatomy, and I'm not going to go through this in great detail, but just to give you a sense, the heart is made up of 4 chambers. There's the left, and we can think of this, and I'll go through this, but there's a left side and the right side. So there's a left ventricle here, left atrium here. It pushes blood out the aorta, which is this big artery, and that is red, and we're going to talk about the color differentiation, but it carries oxygenated blood, the aorta, to all the arteries that deliver oxygen to our tissues. Then it returns through these blue vessels. They're veins, right? So it's got deoxygenated blood in it. It dumps that into the right atrium, the right side of the heart, dumps it into the right ventricle, which then pumps it to the lungs, goes to the lungs, and then comes back to the left side of the heart. And I'm going to go through this more in detail. But basically 4 chambers, incredible design, allowing our blood or our oxygen to be delivered to our tissues. So the cardiovascular system includes the heart, but it also includes that enclosed network of blood vessels, arteries and veins. So by convention, again, arteries are red. So on this slide, you can see the left side of the heart, and red comes out. All the vessels coming out of the left side of the heart are red. What? They're oxygenated. So by convention, red blood cells, and this bears reality, which I'll describe, but then venous blood, which returns from the tissue, so the blood goes all the way out into these arteries, delivers its oxygen, and then is deoxygenated and comes back in these blue vessels called veins, okay? Now, there are 60,000 miles of blood vessels in an average human body. 60,000 miles. So again, this convention, this color convention, where you see arterial blood, deoxygenated blood, and deoxygenated blood being blue, that actually bears some reality. So in cardiac anesthesia, when my surgeon colleague is trying to kill a patient, just kidding, and we have to bring a patient back because they're bleeding after cardiac surgery, sometimes oxygenated blood is bright red. So the arterial blood is bright red, and that's true in reality. Deoxygenated blood, venous blood, is dark red. And if it's really deoxygenated, it's blue. You've seen people maybe that are dead or near death that are blue in color. Truly, blood turns to a dark red and almost blue tinged color. So that is valuable information to me as we open up a chest and we go, where's the bleeding source? Well, it looks like it's there. It's dark blood, probably venous, versus bright red, meaning arterial. So it can be helpful. That was an aside, sorry. So you can think of the cardiovascular system as sort of two subsystems. And here's what I was talking about, the right side and the left side. So the left side of the heart gets blood from the lungs, it's oxygenated, it's red, goes into the left ventricle, and then the left ventricle pumps all that blood out to the tissues, goes through capillaries, oxygen, CO2 is exchanged, and then the venous blood returns to the right side of the heart, where it's then pumped back to the lungs, and the circuit continues. All of our tissues have very small little vessels called capillaries. And again, in these capillaries is where that oxygen is delivered and the CO2 is picked up. So these are our tissues. Blood comes in on the red side carrying oxygen, delivers its oxygen, picks up CO2, goes back into the veins, and then goes back to the heart. The RBCs have to squeeze through these capillaries, and it puts them in very close proximity. Here's that same slide. So the RBCs are really close to the tissues, right? And that close proximity allows the oxygen and CO2 to transfer efficiently to the tissues and into the blood. RBCs, as I mentioned, frequently have to squeeze through very small capillaries. Many times these capillaries are actually smaller in diameter than the red blood cell itself. So another amazing thing about red blood cells is their design. I haven't proved design yet, but I hope you'll let me use that term. But their design allows for what's called deformability. Their design allows the RBC to alter their shape, squeezing through these very small capillaries, and again, putting the RBCs in very close proximity to the tissues where that gas exchange can take place. So when they leave those capillaries, then they regain their normal Kalachi shape, right? And so these are some interesting slides. The capillary, you can see, sometimes they call them bullet-shaped. The RBCs actually kind of take the shape of a bullet. They have to squeeze through the capillaries. And here's a microscopic image of a red blood cell coming out of a capillary. You can see it's sort of a squash donut or a squash kolache. So now let's talk about hemoglobin. It turns out, we talked about hemoglobin coming into blood, it turns out oxygen is not very well dissolved in the blood. So hemoglobin is the molecule that's inside the RBCs that allows oxygen and carbon dioxide to be carried efficiently. Without hemoglobin, our blood could not carry anywhere near the amount of oxygen to our tissues to supply for our needs. So in this first slide, hemoglobin is made up of 4 polypeptides, 4 proteins, 2 alphas, 2 betas, and to those, an iron molecule is bound, and this is called a heme group. Each of those 4 heme groups can bind 1 molecule of oxygen. So each hemoglobin molecule can bind 4 oxygen molecules. Now, there are hundreds of millions of hemoglobin molecules in every RBC. Hundreds of millions of these in every RBC. So that's a lot of oxygen carrying capacity, right? So now remember, just as a review, we breathe lungs into the alveolus, the oxygen comes out, carried by the RBCs to the tissue, oxygen gets dispersed to the tissues where it's needed for cellular respiration, CO2 is taken up, goes back to the lungs, and exhaled. Just a review. Because when I gave the talk before, they were like, you need to bring that together better. I hope that did it to some degree. So another amazing thing about hemoglobin is it's seemingly designed for its important role. Designed, again. And this is described by all you people, docs out here. You remember this? Let's talk about the oxyhemoglobin dissociation curve, right? Shift to the left, shift to the right. No, we're not going to do that. But I think it's important to realize that hemoglobin is amazing, and this curve allows for efficient transfer of oxygen CO2. What I mean by that is, so when the hemoglobin is deoxygenated, and maybe is carrying CO2, it's got this conformation of shape called unbound conformation or shape. And so when the hemoglobin hits the alveoli and the first oxygen molecule binds to it, a shape change occurs in the hemoglobin, and this shape change increases hemoglobin's ability to bind more oxygen and to get rid of its CO2. When the RBC reaches the tissue, now it's holding oxygen, another extraordinary thing happens. The loss of the first oxygen molecule to the tissues causes another shape change in hemoglobin, which allows it to release more of its oxygen avidly and pick up more of that CO2 that it then returns to the lung. So hemoglobin is an extraordinary molecule, and I believe is finely tuned to serve its function. But again, according to standard evolutionary theory, there is no such thing as design and fine tuning. So how does the body regulate the delivery of oxygen? When we exercise, for example, the muscle cells need more oxygen, right? When we eat a big, maybe you ate today, your gastrointestinal cells need more oxygen to do what they need to do to digest in that circumstance. Now, I'm not an engineer, but when looking at the regulation of these physiologic systems, I think it's sometimes helpful to consider engineering terminology. There's sensors, regulatory control centers, and effectors. So think about your thermostat in the house or in here, right? That's the control center. It's got sensors, thermostat or thermometers out there somewhere that tell the control center what the temperature is. So if the sensor says it's too hot in here or too cold in here, it transmits that information to the control center. The control center then sends out a signal to an effector, which is either in this case your air conditioning or your heater, and that effector takes action that then changes within fine parameters, what your thermostat is set for, to keep normal values. So you can think of it physiologically the same way. There's control centers, there's sensors, there's effectors, in order to keep our homeostasis, our normal values within those normal ranges. Biological systems, in fact, are finely tuned with tolerances that are very small. Now, we don't understand in physiology how the system knows what a normal range is in most physiologic systems, but it's not designed. So let's briefly look at the regulation of oxygen flow. It starts with breathing, right? We talked about this, bringing oxygen into the body. How is our breathing controlled? Well, you can control your breathing voluntarily. You can hyperventilate or you can hold your breath, but the majority of the time, your breathing is controlled involuntarily. So in slide number 1, again, thinking in engineering terms, there are sensors in various locations. For example, the muscle. Now this slide's got a lot of activity. I'm just going to focus on this. There's a sensor in the muscle that sends an electrical signal to our respiratory control center in our brain, in the medulla. So that signal, when your muscle increases its activity, even before your muscle says, I need more oxygen, there's an electrical signal sent to the respiratory control center that says we need to start bringing in more oxygen. And so what's the effector? The respiratory control center then sends effectors to the muscles of breathing, if you will, the diaphragm, the intercostal muscles, increase your, what we call minute ventilation, increase the amount of air that you bring in to your body. So an amazing system. So there are also chemoreceptors, which we don't understand how they work, but I mean, mechanistically, we don't understand how they work. But these sensors that are located in the brain, the aorta, and the carotid arteries. So the brain has chemoreceptors. The aorta, which is, remember that vessel I pointed to just coming out of the heart, and your carotid arteries, they have chemoreceptors that sense oxygen, carbon dioxide levels, and pH. So if oxygen is low or carbon dioxide is high, remember we talked about oxygen, CO2, those chemoreceptors again send a signal to the respiratory control center that says we need to increase breathing. And so if our oxygen level's low, your body says involuntarily, let's pick it up, guys, let's breathe more. Did that make sense? Okay. So, and now again, these systems are finely tuned. How they're tuned, we don't exactly know. We know they work and we know how they work, but mechanistically, we don't know how they work. Again, it's sort of a Goldilocks thing. You remember Goldilocks? The porridge, too hot, too cold, just right. It's kind of same with the thermostat, right? Goldilocks thing. You can't be too hot, too cold, you got to be just right. Same here. Oxygen levels, if too much oxygen is actually toxic. Too much CO2 can be toxic. So just the right amount, not too hot, not too cold, just right. You'll hear me use that Goldilocks phrase, so now you know what I'm talking about when I say that. And again, this fine-tuning is amazing, but according to standard evolutionary theory, it's not designed. I always like to throw scriptures in. I love this one about breathing. I will make breath enter you, and you will come to life, Ezekiel 37:5. Once oxygen is brought into the lungs and transferred to our RBCs, how does the body control the delivery of that oxygen to our tissues? It's not enough to just merely pump the blood around, right? The cardiovascular system has to adjust blood flow through blood pressure in a lot of circumstances with changing needs of different body tissues. So again, if your muscles are working hard, you got to have more blood and more oxygen going to your muscles. Same with all of our other organs or our other tissues. So the cardiovascular system has to be controlled, and it sort of has two regulatory systems that I'm going to discuss. One is a sort of rapid control system, and another is sort of a longer-term control system. So as an example, again, this rapid control system, when, let's say you're gardening, you bend over, you do a little gardening work, you get back up, whew, maybe you're a little dizzy, right? Worst case scenario, you faint, right? So the issue there is when you stand up, gravity pushes on your blood volume, okay? And so that gravitational force, as you elevate yourself, can decrease the blood flow to your right heart, decrease the return of blood to your right heart. That can compromise your blood pressure, right? And so if that were to happen and you didn't have instantaneous regulatory control, you would faint every time you stood up. So again, thinking somewhat in engineering terms, there's sensors. Now, remember I said there's chemoreceptors, chemical sensors in the aorta and the carotid. Same thing. We have what are called baroreceptors. We have receptors in the carotids and the aorta and carotids that sense blood pressure. So when those arteries, when you stand up, blood pressures drop and instantaneously send a signal to the cardiovascular control center in the brain, which then sends effector signals out to the heart and to our blood vessels. So what do they do to the heart? Here's the medulla, or the medullary control center. That's a bigger picture of this section right in here. So those signals go out to the heart and they say, whoa, you gotta pump harder. Increases contractility of the heart, increases stroke volume, amount of blood coming out of the heart with each beat. In addition, it sends signals to the blood vessels in your body and says, you need to tighten up because we need blood coming back to the heart. We need to maintain blood pressure. So that's an example of the rapid control system for blood pressure. Now, again, of course, intuitively we know that this needs to be finely tuned, right? Or balanced. That's this balance right here. So if these mechanisms increase our blood pressure, and that's the example I used, and blood flow, if they do that too much, that can be problematic, right? If I increase your blood pressure in your vessels, they have limitations to how much they can take. They can bleed, they can rupture, right? If that happens in your brain, you may have a stroke. So it has to be balanced. It's a Goldilocks thing, just the right blood pressure, not too much, not too little. And again, kept within normal ranges. And again, it's beyond our physiologic understanding at this point as to how exactly the body knows what the normal range is in those circumstances, but it's not designed. I hope you know I'm saying that sarcastically. Okay. So our bodies also have long-term mechanisms to control blood pressure. So not just this instantaneous mechanism, but over time. So I'm going to try and go through this. It's interesting, but I'm going to whip through it. My wife said I shouldn't whip through this one because she thought it was interesting. So it's interesting, the kidney does a lot of stuff more than make urine, right, and filter our blood. The kidney has these baroreceptors in it too. So it senses blood pressure. If it says, for example, the blood pressure is low, it secretes an enzyme called renin. Okay, what does renin do? The liver secretes an enzyme called angiotensinogen. Renin converts angiotensinogen to angiotensin 1. Then, surprisingly, there's an enzyme in the lung called angiotensin converting enzyme. It predominantly is produced by the lung. It converts angiotensin 1 to angiotensin 2. Now, angiotensin 2, well, what does it do? It affects the blood pressure in a couple of ways, and these are the two most important. So angiotensin 2, remember we talked about increasing the vasoconstriction of the blood vessel, increases your blood pressure. Angiotensin 2 is a potent vasoconstrictor. So it increases your blood pressure by squeezing your vessels. What else does it do? And again, this is a long-term thing. It's not an instantaneous thing because it's a chemical way of communicating, not an electrical via your nervous system. Angiotensin 2 also causes your adrenal glands to secrete this hormone called aldosterone. What does aldosterone do? Goes back to the kidney and says, hey, we need to keep more water in the system. So it reabsorbs sodium. Along with sodium comes water. Well, what does that do? Well, if you keep more water in your blood and not fever as much, you have more blood volume. If you have more blood volume, you potentially have a higher blood pressure. So again, controlling the blood pressure. There's another mechanism which I'm going to briefly discuss. Similar, but we have a regulatory control system in the hypothalamus that actually senses what's called osmolarity of the blood. That just means basically how thick it is, how concentrated it is, right? So sometimes because dehydration can contribute to low blood pressure, right? So if you're dehydrated, your blood's going to be concentrated. You don't have enough water in it. So what does the hypothalamus do? Well, first of all, it says you need to drink some more. It says you are thirsty. And so you start to drink more water, or hopefully you do if you get that thirst sensation, right? At the same time, the hypothalamus goes, all right, we need to do some more. So I'm going to bring, again, more water back into the blood. So it releases a hormone called antidiuretic hormone, ADH. Diuresis, you know what that is? You take a diuretic, what do? You urinate a lot, right? So you're allowing that, that water to be removed from your system. So what does this do? Antidiuretic. It says, nope, I'm holding on to the water. Again, what does that do? Increases your blood volume, which can increase your blood pressure. ADH. So ADH, antidiuretic hormone, practical application of where that comes into play. Alcohol. You drink a little alcohol, all of a sudden, why do I have to go to the bathroom so often? It blocks ADH. It says, I'm going to block ADH. So instead of being an antidiuretic, you diurese. So that's the mechanism whereby when you drink alcohol, you may drink a beer, boy, got to go to the bathroom. You think it's just the volume? No, it actually dehydrates you because it blocks ADH. That was probably more than you wanted to hear. So, now again, in high blood, I described sort of high blood pressure situations. In low blood pressure situations, these mechanisms are basically working in reverse. And again, a Goldilocks thing, got to have the right blood pressure. Not too high, not too low, just right. So as we well know with age, sometimes these mechanisms to control our blood pressure aren't working as well. Blood pressure medicine? Yep. Okay. So sometimes if the mechanisms get a little out of whack, you may need some pharmacological help. Now I'm a doctor, so I go to pharmacology versus natural. But my point is sometimes you may need some treatment to keep blood pressure under control because you don't want to be too high. The most common blood pressure medicines that we use today act on these mechanisms. So there's one called a angiotensin receptor blocker. So basically, it blocks what angiotensin II does. It blocks primarily this vasoconstriction. So that angiotensin receptor blocker blocks this, hopefully keeping your blood pressure down because it's not going to cause vasoconstriction. There's also an angiotensin converting enzyme inhibitor. So it prevents the conversion of angiotensin I to angiotensin II. So again, angiotensin II can't cause vasoconstriction and increase your blood pressure. Again, these are the two most commonly used today. Blood pressure medicines come and go, but they're the most commonly used today blood pressure medicines that we can use to regulate our blood pressure. We have learned recently that I find fascinating, so I'm going to go through it. It was a paper published just in 2021 describing this incredible, what's called signaling network. Recall, I said the kidneys have baroreceptors. They can sense blood pressure. But for a long time, we didn't know, and then that blood pressure, of course, they release renin if the blood pressure is low. If blood pressure is high, they don't release renin. Because remember, renin works in that cascade back here that causes angiotensin to go to angiotensin II and then causes vasoconstriction. We didn't know exactly how this mechanism worked. I want to read first the sort of summary statement that they make. These studies show that the enigmatic baroreceptor, that's the receptor I was talking about in the kidney that senses blood pressure, is a nuclear mechanotransducer that resides in the renin cells per se and is responsible for the sensing and transmission of extracellular physical forces, that's the blood pressure, to the chromatin, that's the genes, in the renin cell. to regulate renin gene expression, renin bioavailability, and homeostasis. So this is what's basically going on here, and I'm going to try and be brief. So remember I said blood comes into the kidneys, perfusion pressure, blood pressure. It goes into, this is the glomerulus. This is where blood is filtered. So blood pressure is sensed by these renin cells right here, these yellow, all these juxtaglomerular cells they call them, renin cells. Well, how does that happen? Well, around the renin cells is this extracellular matrix. Think of ECM. Think of that as just the stuff around your renin cells. Well, it turns out when blood pressure comes in, this extracellular matrix, depending on the blood pressure, it wobbles. Okay? So how much wobble? Dependent on the blood pressure, right? But the amazing thing is now, it turns out, we found out that there's this protein called integrin that when this wobble occurs, that wobble, that mechanostimulation, is transferred inside the cell of the renin cell, and it transmits that mechanical stimulation to another protein called actin, okay? And then actin transmits that same wobble, that same pressure, inside the nucleus. This is the nuclear membrane of the renin cell. It transmits that same mechanical force inside the nucleus, which affects the chromatin and gene expression, which we have no idea how, but it affects how genes for renin are produced. So if the blood pressure is high, you get low renin production, right? If the blood pressure is low, you get high renin production, and this is the mechanism, which is extraordinarily elegant as to how that takes place. So again, how these cells are calibrated, how the cells know how much renin to release or not to release based on the amount of wobble in that extracellular matrix. But it's an extraordinary example, in my opinion, of a biomechanical cellular mechanism. And to many scientists, like myself, I struggle to see how chance, random events could be responsible for such elegance. Of course, the authors of this article made this statement. Renin expressing cells are essential for survival, perfected throughout evolution to maintain blood pressure. So I think of it as sort of a all hail Darwin expression. In this study itself, they in no way discuss potential evolutionary mechanisms for this. So this statement demonstrates sort of the unquestioned, in my opinion, premise by which many scientists approach biology, right? Evolutionary theory. There is no design. I think of it this way. Remember Obi-Wan Kenobi in Star Wars? He was on the, what are those things called? Hovercraft, right? With Luke Skywalker, and he had R2-D2 and C-3PO in the pack, right? And they were looking for those two droids. So he came up to that stormtrooper, and he asked about the droids, and Obi-Wan goes, "These aren't the droids you're looking for." And the stormtrooper goes, "These aren't the droids we're looking for." So think of it in the same way. This isn't the design you're looking for. Kind of hand-waving. Anyway, I thought it was a great analogy. So, okay, let's take a quick look at red blood cells and hemoglobin. I'm going to press on rapidly because I know John's starting to get twitchy over there. Your body produces 20 billion red cells a day. Red blood cells, remember? We talked about them, oxygen carrying capacity. That's 2 million every second. So how does the body control this process? How many red blood cells? How much oxygen do we need, but not too much? Again, Goldilocks. Just the right amount of RBCs, just the right amount of hemoglobin. So it turns out in the kidneys, again, right? They're a versatile organ. They, in fact, have cells in them that have chemoreceptors. They sense the amount of oxygen in the blood. When the kidneys sense the amount of oxygen in the blood is low, they secrete a hormone called erythropoietin, EPO. EPO, ring any bells? Austin native, got in trouble with EPO, Lance Armstrong, okay? So maybe some people believe he didn't take it. He admitted it, right? So erythropoietin, when the oxygen is low in the kidneys sense that, it releases a hormone called erythropoietin that binds to what are called stem cells in the bone marrow. Stem cells are like undifferentiated cells. They can produce red blood cells, white blood cells, a variety of things. So this erythropoietin binds to these stem cells, erythropoietin on the surface of a stem cell, and through genetic regulation turns that or marks that stem cell to become a red blood cell. Then that stem cell We're going to talk about hemoglobin, but the stem cell becomes a red blood cell, which goes back into the blood. So erythropoietin, low oxygen in the kidney excretes erythropoietin, stem cells turned into RBCs, more RBC production, more oxygen carrying capacity. Again, these are specific receptors, which why they're just on those cells, we don't know. And why when they're marked as to become red blood cells, they develop other surface-specific receptors, we don't know. Going fast. So, briefly, another amazing regulatory system, iron. Remember, iron's needed. Remember, it's inside a hemoglobin. So RBCs need iron to create hemoglobin. But there's an issue, too. It can be toxic to our organs throughout our body. So, again, Goldilocks, right amount of iron, not too much, not too little. The body makes about, again, 20 billion red blood cells a day. So the right amount of iron needs to be brought into the body to make the hemoglobin for those 20 billion cells. Now, the liver stores iron, and I don't know, I used to go to, my grandma loved Luby's, so you go to Luby's and she gets the liver and onions, and I'm like, I do know. It's a good source of iron. Turns out grandma's right. Iron is, in fact, the largest store, the liver is, in fact, the largest store of iron in our bodies. And the liver also monitors the iron levels in our bodies. We don't know exactly how. So depending on the amount of iron stored in the liver releases a hormone called hepcidin. Hepcidin is this blue guy right here, released from the liver. And that hepcidin acts on a receptor in your duodenum called ferroportin, which is FPN. So the hepcidin acts on this receptor in your duodenum. When you eat iron, my grandma, liver and onions, right? Iron gets absorbed into these duodenal cells, and based on hepcidin's activity on this receptor, iron's brought into the bloodstream, okay? The liver also produces a transport protein called transferrin, because free iron is toxic to your body. So it needs to be safely transferred to where the iron needs to be utilized. So the liver produces this transferrin, which then, again, remember we said RBCs are marked in your bone marrow, or stem cells are marked in your bone marrow to become RBCs. So this transferrin's got to transfer that iron to those RBCs so that those RBCs can make hemoglobin. Here's transferrin and iron. Again, a specific receptor on the surface of the stem cell that's marked to become an RBC. Transferrin and iron are bound to that receptor. Iron's brought into the cell, so hemoglobin can be made by the RBC. How this extraordinary system came to be and how it's finely tuned to provide us not too much, not too little, but just the right amount of iron for hemoglobin production is not designed. So I've given you, sorry about the physiology, I hope that wasn't too much, but I've given you an overview of some basic physiology related to the delivery of oxygen to our body's tissues. Now, I know some of it was a little complicated, but the simple act of breathing in oxygen and getting that oxygen to our cells is extraordinarily sophisticated. There are many other physiologic systems in our body that are more complex and maybe even more amazing than what I've described in the systems I presented. As I mentioned previously, my ultimate goal wasn't to have you understand these physiologic systems, but rather to make the case that with intellectual honesty, these systems might best be explained by design. So again, what's the debate? The debate is causation. Is the human body designed or a product of natural processes? Evolution versus intelligent design. Moving forward, you saw I just ran through a lot of physiology. Well, when it comes to this argument between design and evolution, I'm going to use an appeal to authority, right? Because I don't want you to stand up. I mean, hopefully you can believe me about the physiology, but when it comes to the debate, I need some other experts to make comments. So you're going to see me quote a lot of experts in the field. Appeal to authority. It's not just me saying it, it's these guys saying it, okay? So these authors, Lofman and Glicksman, wrote a book, Your Designed Body, some years ago. I think they did a pretty good job of making, in this statement, they said, modern science in both biology and medicine is pretty good at discovering and describing the body's parts, their interactions, and their processes. But it falls short in explaining how these things could have come into existence. Current explanations for from evolutionists consists largely of imaginative storytelling and fails to answer any of the salient questions posed by a detailed analysis. Remember Charles Darwin, Origin of Species? There's another quote in there that I think is relevant to our conversation. He said, "If it could be demonstrated that any complex organ system existed which could not possibly have formed by numerous successive slight modifications, my theory would absolutely break down." I appreciate your transparency on the weakness of your theory, Charles, and I think maybe some have come to see that it is breaking down. So with that in mind, I want to touch on just two real simple concepts, I think, when it comes to design in biological systems. The first is this idea of irreducible complexity. Michael Behe sort of used this and created this concept when describing complex biological systems. He said, and he uses the mousetrap as an analogy. He said, a single system which is composed of several interacting parts that contribute to the basic function, and where the removal of any one of the parts causes the system to effectively stop functioning, that's an irreducibly complex system. So again, a mousetrap, it's got a base, a catch, a hammer, a spring, and a holding bar, and all these parts are necessary for it to function. So if a mousetrap is considered like a biologic system, all the parts of that biologic system, like individual proteins, would evolve sort of separately. But why would these parts, this base, this hammer, why would they evolve if they have no function until they're put together? And so if there's no function, there's no increase in fitness, so that can't be passed on to future generations. So when considering some of the physiologic systems I've described, many scientists contend that they are irreducibly complex. Our own Fazal Rana, president of RTB, wrote this book, The Cell's Designs. He says this: irreducibly complex systems cannot be produced from protosystems by the Darwinian process of slight incremental change that inch towards the finished product. Any protosystem that lacks even one of the parts that contributes to the basic function is non-functional. Without function, natural selection has nothing to select. So the physiologic systems I've described are interdependent. I said that before. They all work together, and I showed you how all the kidney, liver, cardiac, they all sort of work together, and I would contend that they are, in fact, irreducibly complex. To get oxygen into human tissues, the respiratory system is dependent on the cardiovascular system, which is dependent on red blood cells, which is dependent on hemoglobin, which is dependent on iron metabolism. So all these systems require regulation within tight parameters, or the systems are non-functional and therefore irreducibly complex. Again, these authors said, could these systems be assembled gradually? How did many interdependent control systems get their start? Where did the parts come from? How were all the parts properly assembled? Where did the assembly instructions come from? How was the whole finely tuned for just the right capabilities with just the right capacities? If a particular system is necessary for survival, how could an organism build it when it requires that system just to be alive? Michael Behe said it this way. He said, and with regards to the science behind irreducible complexity, he said, there is no publication in the scientific literature, in prestigious journals, specialty journals, or books that describes how molecular evolution of any real complex biochemical system either did occur or even might have occurred or evolved. So how could these interdependent, irreducibly complex human physiologic systems be evolved? That question is really not being asked. It's usually just a de facto premise. This isn't the design you're looking for. Sorry. So the second idea I want to briefly discuss is this idea of systems biology. And in the past 15 years or so, scientists have begun to study biological systems with sort of a new approach. And it's called the systems biology revolution. Essentially, engineers have started to work with biologists using engineering models to look at biological systems. These engineering models are essentially a design-based model. So if systems biology is using design-based models to study biologic systems, one can see how that could be a challenge to the standard evolutionary theory, because design is not a part of the standard evolutionary theory. These authors make the following statement with regard to systems biology. A hope for understanding complexity in biology is to uncover through a calculus of purpose by asking teleological questions, such as why cellular networks are so observed given their known or assumed functions. So teleology is the ID argument, right? That there is purpose and design in what we see in biological systems. So what they're saying is one needs to assume design and consider purpose to make progress in the understanding of biological systems. So again, a challenge to standard evolutionary theory, which states there is no purpose and no design. Here's another publication in 2016. As a discipline, systems biology shares many characteristics with engineering. We identify the cells as an embedded computing system, and as such demonstrate that systems biology shares many aspects in common with computer systems engineering, electrical engineering, and chemical engineering. So what scientists are seeing is that the same systems layout we see in human engineering is consistent with what we see in biological systems. So from a big picture perspective, biological systems show engineering design. So the evolutionary biologists are now in fact being forced to accept the fact that science, especially in biological systems, points to design and purpose. But they also understand the implications of that. Here's a publication this year called, I didn't talk agency in biological systems. It's almost like they're giving identity to biological systems. They can create their own, they can guide their own future. I didn't have enough time to talk about that, but the book is titled Organisms as Agents of Evolution, and they make this statement. There is a common suspicion that talk of goals and purpose renders biology teleological, thereby raising notions of design and of some overarching plan. Invoking agency as a real biological phenomenon seems to some to open the door. Notions of purpose and goals might invite quasi-religious explanations of phenomenon in the manner of intelligent design. I would agree. The door is now open. Michael Behe said it simply, the strong appearance of design allows the simple argument. Design should not be overlooked simply because it's so obvious. How's everybody doing? So, I'm an anesthesiologist. I do a pretty good job of putting people to sleep, and I get paid for it. But I'm also supposed to wake you up. So, stay with me. This next part is, I think, the most interesting stuff, and it's where the challenge is to the standard evolutionary theory. So, John and Lacey, David and Deborah and I had dinner with Fuzz, again, the president of RTB, not too long ago, and we discussed briefly, John ignored me, discussed some topics with Fuzz about my thinking about this presentation. And he made what I thought was a surprising statement. And I put this quote in red quotations because I don't remember exactly what he said. But basically he said this: most evolutionary biologists who understand the big picture of evolution aren't neo-Darwinists. So this is a big deal, right? And he understands what science is thinking nowadays. But the evolutionary perspective has framed how a large percentage of our population looks at the world and looks at nature. It has defined a worldview that sees the universe and the earth and life, including humanity, as just some sort of cosmic naturalistic accident, which of course, as I mentioned before, has, I think, significant moral and theological implications. So if in reality, as Fuzz said, those who understand the big picture of evolution don't believe the standard evolutionary theory, then it certainly opens the door to consider other options. So most people, I think, at least in my mind, historically, I've always thought of the scientific consensus as being the standard evolutionary theory. Well, it might not be. We're going to show you some stuff. So it turns out I think Fuzz is right. And this is not certainly common knowledge, at least in the general populace. This is an organization called The Third Way, and you can look it up on the website. This is sort of one of their statements. They say, the DNA record does not support the assertion that small random mutations are the main source of new and useful variations. We now know that the many different processes of variation involve well-regulated cell action on DNA molecules. Moreover, some neo-Darwinists have elevated natural selection into a unique creative force that solves all the difficult evolutionary problems without a real empirical basis. many scientists today see the need for a deeper and more complete exploration of all aspects of the evolutionary process. So these guys are scientists, many of them National Academy of Science members. They are in the consensus of science, in the scientific community. They're not design proponents necessarily. They're just saying the standard evolutionary theory is insufficient. So science apologetics organizations, including R2E and the Discovery Institute, the advocates for intelligent design, I think initiated some of these challenges that have come to the surface. But more recently, the challenges are coming directly from the evolutionary science establishment. Many scientists are now calling for a revision of the evolutionary paradigm. These scientists, again, not creationists, not intelligent design proponents, just saying we need a new look. And as a side note, I like this name, the third way. I don't know how they came up with it, but so what are the first two ways? Well, I think they're standard evolutionary theory, and I think they're giving a nod to the challenges of intelligent design. And so they're the third way. They're not buying either of them, but they're going to find a third way. But again, I find and I appreciate the fact that the organization may be respecting the intelligent design findings. So here's a publication in 2015 which sort of shines light on the issue. In recent years, we've seen increasing numbers of biologists who are dissatisfied with conventional neo-Darwinian evolutionary theory. Biologists are also unhappy with the resistance of an entrenched scientific establishment to considerations of new ideas. Part of this resistance is owing to the fact that the establishment has taken on a kind of siege mentality owing to assaults from the intelligent design community. So evolutionary biologists of the past would never have imagined this happening, right? So evolutionary biologists in their own camp are now questioning the standard theory of evolution, and in fact acknowledging the arguments of intelligent design. And then came a meeting, prestigious meeting, the Royal Society in 2016. The title of that meeting was New Trends in Evolutionary Biology. The take-home message, briefly, is that standard evolutionary theory, again, is insufficient to explain the large-scale transformations we see in nature. So, and this would include the complexity of the physiologic systems in the human body that I described. So to reinforce, and I'm sorry about this long-winded quote, but this article came out in 2017. Gerd Mueller, very well-respected evolutionary biologist, very well published, he made this statement. He said, current evolutionary theory largely avoids the question of how the complex organizations of organismal structures, physiology, development, or behavior actually arise in evolution. A rising number of publications argue that a major renovation or even a replacement of the standard theory of evolution, indicating that this cannot be dismissed as a minority view, but rather is a widespread feeling among scientists. Indeed, a growing number of challenges to the classical model of evolution. Sometimes these challenges are met with dogmatic hostility. The real issue is that genetic evolution alone has been found insufficient for an adequate causal explanation of all forms of phenotypic, evolutionary term, complexity. So of all forms of complexity, not only of something vaguely termed macroevolution. So macroevolution, we're going to discuss that. So macroevolution, it's a question of micro versus macroevolution. Some of you have heard this conversation before, but microevolution is basically a process by which an organism can change or adapt in sort of small ways. For example, in medicine, we see bacteria that develop resistance to antibiotics. That's a small change. And without argument, the mechanism is in fact associated with what we could consider standard evolutionary theory. That is, mutations can occur in bacterium that can cause them to develop a resistance to certain antibiotics. So, microevolution. But macroevolution, on the other hand, refers to larger evolutionary changes that result in complex biological systems or, in fact, new species. So if we look at Darwin's tree of life, right, the single organism advances along the evolutionary tree and develops into more complex life forms. Sometimes sarcastically people say, from the goo to the zoo to you, right? So generally, most would agree, with intellectual honesty, there is scientific evidence for microevolution, bacterial resistance to antibiotics. Mueller, this author that I just quoted, is only claiming that those same mechanisms are insufficient to explain macroevolution. To reinforce editorial, just in 2021, James Shapiro, a very well recognized evolutionary biologist, lots of publications, he said this, referring to the Third Way, that organization formed back in 2014. The Third Way of Evolution was founded in 2014 to make the public aware that contemporary evolution science is not limited to the neo-Darwinian modern synthesis of the past. This was important to do because evolution was challenged as incapable of explaining biological complexity by the intelligent design movement. Modern synthesis was based on Darwin's preference for gradualism, to elevate natural selection as the sole force determining the direction of evolutionary change. By asserting that the accumulation of random slight variations was the basic mode of both short-term, micro, and long-term, macro, evolutionary change, the modern synthesis also ignored the distinction between microevolution and macroevolutionary origination. In so doing, modern synthesis failed. So, I think design arguments are having an impact. Science is having to look at it. And here's another publication more recently from Biophysics and Molecular Biology, 2022. Selection by survival of the fittest was a captivating idea. There has been limited progress to the modern synthesis. Now there is evidence to document that selection based on survival of the fittest is insufficient for other than microevolution. So, standard evolutionary theory, which has been the educational indoctrination for, I would say, 3 generations. It looks like maybe there's some challenges. It may not be correct. So how long until those challenges to the standard evolutionary theory are revealed in our biological education? I hope soon, but I'm not holding my breath, right? So, if the standard evolutionary theory is insufficient to describe complex biological systems, how do we move forward? How does science move forward? Max Planck, theoretical physicist, he won the Nobel Prize in 1918. I think he made this statement well when it relates to this. A new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because its opponents eventually die. A new generation grows up that is familiar with it. So paraphrased, many people say, science advances one funeral at a time. So it may take some time, but the scientific community, I think, is showing movement in the right direction of a new truth when it comes to evolution. My suspicion, though, naturalistic theories will look more and more like design, but will be attributed to materialistic mechanisms. At some point, design, and even a designer, may be undeniable. Last few slides. Anthony Flew, philosopher. He was considered at one time to be the world's most notorious atheist. He published numerous books and participated in debates espousing atheism and using science as one of his principal arguments. He was known for the statement, an honest person must follow where the evidence leads. He wrote a book called There Is a God. He said this, I have to go where the evidence leads. I think the most impressive arguments for God's existence are those that are supported by recent scientific discoveries. The argument for intelligent design is enormously strong, stronger than when I first met it. The findings of more than 50 years of research have provided materials for an enormously powerful argument for design. So it's no surprise to many, so it was surprising to many, especially in his former camp, that he changed his opinion. And basically his book, titled There Is a God, expressed his new perspective that the evidence for design pointed to God. He goes on, I now believe that the universe, life, and reproduction originate in a divine source. This is the world picture as I see it, that has emerged from modern science. My discovery of the divine has thus far been a pilgrimage of reason. So not surprising, those on the other side criticized him for his new philosophy. Nonetheless, I find it encouraging that design is becoming a viable, intellectually honest option, even amongst mainstream scientists. So I think, this is a great finishing scripture. For since the creation of the world, God's invisible qualities, his eternal power and divine nature have been clearly seen, being understood from what has been made, so that people are without excuse, from Romans 1, 20. So being understood from what has been made. What is made is designed.
All right, so I take back what I was saying about you being intelligent, because, I mean, we all knew that, right? No, that was very, very good, very informative. Really enjoyed that. So, want to get to some Q&A, and so just real quick, rules for the Q&A. If you have a question, raise your hand, and either David or myself will bring a microphone to you so that you can ask your question. We do ask that you ask a question and not just ramble on with commentary, and we go 5 or 10 minutes and there's not even a question. Again, other people have questions. If you need to set up your question with a little bit of commentary, then please do so. But at the end, let's ask a question and move on so that other people can also get to their questions. And secondly, I'm the president. I get to ask the first question. So, as I was watching your presentation and I was trying to think from an atheist perspective or from a Darwinist perspective, what I would like to ask, I think one of the things, even though you do a great job of illustrating fine-tuning and design, one of the things particularly Richard Dawkins likes to argue is that there are systems that appear to be poorly designed. And for instance, the eye, even though he admits on the one hand it's this exquisite precision instrument, on the other hand, quote, it was designed by a complete idiot. Okay, end quote. So, are you familiar with some of the arguments of poor designs, and what would your response be to those?
Yeah. So, it's certainly interesting, and these have been around forever, and books have been written, and people have made their careers out of sort of espousing evidence of poor design, especially in biological systems. But when those systems are looked at in more detail, sometimes what appears to be design characteristics that might have been improved by a designer, frequently it turns out that new physiologic findings, new anatomic features, maybe even new physics determine that those designs actually turn out to be, okay, well, maybe they could be a little bit different, but what we complained about turns out to be a good feature of that design. So, I mean, you go into detail and I could spend a lot more time, but yes, I'm aware of those. But for the most part, I think the apologetics community has done a pretty good job of sort of quashing those arguments to a large degree.
Excellent. Any hands? David? Oh, I'm sorry. Denton.
Can you hear me?
Yeah.
First of all, great talk. Really appreciate it. Really enjoyed it. I like the way you brought scripture verses about blood into the complexity of blood. So my question is, I'm always amazed when you get to go into the doctor, you do the blood chemistry test, and you have all these high and low ranges. You have 30, 40, 50 different tests that are done. How important is that as far as complexity, those ranges? Where do they come from, and how important is that deviation as far as survival or complexity?
Yeah. So I'll answer the second question first, and that is, how important are they? They are extraordinarily important. So, for example, a normal potassium is, depending on the lab, is 3.6 to 4.5, let's say, milliequivalents per liter. And so if you come in and your potassium is in the 5 or 6 range, holy smokes, that can cause huge disruption to your physiologic systems. But milliequivalents per liter, that is an extraordinarily small amount of potassium, right? So they're extraordinarily important. And your second question, where do they come from? Somehow the body knows that that's the amount of potassium or sodium or calcium that we need in our system. So from a physiologic standpoint, where they come from, we don't understand the mechanisms completely. We understand their importance and what those, for example, electrolytes impact if they're out of range, but we don't know how the body determines and keeps those normal ranges. Now, maybe your question was, where do they come from? Well, they come from, as far as the laboratory is concerned, they come from assessment of normal physiologic systems, and there's normal ranges based on that, and that's the way we determine what's normal. Okay.
What keeps the blood cells in in the formation that they are in, like the book, the donut shaped?
Yeah. So there's this cytoskeleton inside red blood cells that, and again, I didn't go through it, but there's a lot of interesting physiology and anatomy, microanatomy, involved in the red blood cells, and the fact that they have that capability. You remember that slide where they're kind of bullet shaped going through the capillaries? Again, allowing their surface area to touch the side of the capillary and therefore transfer the oxygen and CO2 better. But basically there's a cytoskeleton structure inside the red blood cell that with certain external pressures on it can easily collapse into a new formation. So it's kind of like, what was that? A toy that basically you can crumple into a new shape.
Balloon?
Yeah, like a balloon maybe, that you can crumple, but air is holding that thing, of course, but you can push it into a new shape, but then it just regains that shape once it doesn't have that external pressure on it. Yes, sir.
How do you, I know that some biologists think that the most profound thing that happened in life was when, I'm probably pronouncing this wrong, when prokaryotes became eukaryotes, where simple bacteria became complex bacteria, and mitochondria and photosynthesis was delivered, and that was the beginnings of the regulatory system, and from there it was just hundreds of millions of years of evolution, of trial and error, that got where we are today. How do you discount an inconceivable notion to us of hundreds of millions of years of chance that would make us where we are today?
So how do I discount that? Okay, so it's not me discounting it, it's the evolutionary biologist. As I've said, the evolutionary biology camp, the stronghold of current evolutionary biologists, am I in the wrong place? Are discounting it themselves, meaning they're saying that chance events, even over long periods of time, based on what they're seeing in biological systems, cannot account for the extraordinary evolution, if you will, of those biological systems that seem to have and go in the direction of purpose and design and what they sometimes describe as agency. So it just appears that evolutionary biologists themselves are saying the science does not support the fact that these huge transitions could in fact take place. Now, could a bacteria develop a different type of cell membrane which could allow it to be more resistant to heat? Yes, that would be microevolution. But the dramatic changes that we see in biological systems don't appear to follow and be described by that same evolutionary paradigm. If that makes sense. Yes, sir.
Here's a kind of a follow-up
Yeah.
question to that. Are there any identifiable theories that these people are putting forward that are gaining traction? What are the mechanisms of macroevolution that they're talking about?
Right.
Anything?
John made me take that slide out. I didn't have enough time to go through that. But at that Royal Society meeting in 2014, that sort of prompted everybody to start saying, hey, we need to find naturalistic mechanisms other than standard evolutionary theory to describe what we're seeing. And so there's, I'm blocking on his name, I can pull up this slide, but there are a couple. Neutral evolutionary theory is one, and extended evolutionary synthesis is another. There's the two theories that they're positing to replace standard evolutionary theory. They haven't been well studied yet, and certainly, at least from a person's perspective, including some RTB scholars, looking at those theories, they still are lacking, but they haven't been studied. So could they find something in that extended evolutionary theory, neutral evolutionary theory? Maybe. The mechanisms are extraordinarily complex and a little above my pay grade in understanding that. So I rely on people like Annette, what's her last name? AJ, yeah, and Fuzrana. And it appears that those theories are an attempt to integrate design and purpose, but attribute it to materialistic causes, which is going to be tough for them to do. Yes, sir.
That was just great. I'm thinking about a couple of guys, and either they're to be charitable, either like canaries in a coal mine, or like criminals that tell the truth when the police first arrive and then work on their story later. But so either Watson or Crick said it had to be directed panspermia as soon as he saw the complexity of DNA.
Yes.
And even Dawkins, Ben Stein was talking to him and he said, well, we have considered that it could be extraterrestrial. So while we're waiting for the schools to never teach what you just taught us and the old people are dying, I'm thinking if the new, you'll have to put on your tinfoil hat so you can hear me from this point on. Do you think that the new illusion that's going to be peddled is extraterrestrials? Because we're now being told, all these, Navy tapes are coming out. So the next scam will be that life was planted in either, and if you heard this anywhere up in the higher echelons, life was planted here. So we'll either have a middleman between us and God, or the extraterrestrials will be our God.
Yes. So I think that's already happened. To a large degree, and this idea of panspermia is out there, and I think it's an interesting notion, right? Because basically the evolutionary scientists are saying, especially in the origin of life question, right? We have no idea how life began, right? From the goo to an initial bacteria, even an initial life form is so complex. So there has been speculation, theorizing that in fact maybe our planet got impregnated panspermia with DNA, right? And that's how life began. But I think that's already happening. I think if you listen to some songs, ELO, for example, and other movies that are out there, frequently it's at the end, what do you see? A spaceship, And holy smokes, these guys, have brought something profound to the earth. So it's a double-edged sword in my mind, because I think basically by doing that, the evolutionary scientists are saying, we can't explain it. So what's it doing? It's putting it maybe to an intelligent designer. If they don't want to call it God, at least they're saying, there's some design here, but we don't like God, the idea of God being in it. So we're going to say it's maybe aliens, right? So in a way, that's a positive thing. At least that's the way I look at it. Yes.
Thank you for the talk, and you mentioned about Michael Behe and the irreducible complexity. So I'm always confused a little bit. I know Michael Behe he believes in the theory of common ancestry, but he rejects the neo-Darwinist theory. Like I'm always, is it being a design proponent? Can you be consistent with believing in the theory of common ancestry?
So I am not aware that Michael Behe is entirely within the paradigm of accepting common ancestry. I think he accepts lineage ancestry, if you will, meaning subspecies or speciation of common organisms. But I'm not aware that if I threw that Darwinian tree up there, that he would accept that completely. And certainly his teachings and his writings support the idea that evolutionary theory, and irreducible complexity can't be supported by the evolutionary paradigm. So you may know more about Behe's sort of overall evolutionary perspective, but certainly his concept of irreducible complexity is used extensively when the design argument comes up.
Last question right here. Just a quick follow-up on the alien intervention.
Don't hurt me, Richard.
Anyway, just a follow-up on the alien intervention idea. First of all, that is a nod to some sort of intelligent external design, for one thing. But secondly, isn't that just a delay of the obvious? That's a turtles all the way down kind of a theory.
Yes.
Is that right?
Agreed, yes.
Okay, thank you. LD, that was outstanding.